Okay, so thank you for coming to another seminar. Today we have Xesca Reynés. I guess that most of you know her. She's a PhD student in the IMSIS group, and she will be talking about her PhD, I guess, about microbiome, Mar Menor, and things like that. So thank you very much.
Thank you. So yes, as Eneko says, today I'm going to talk from a work that is part of my thesis, which is focused on the study of seagrasses and its associated microbes. The main objective of my thesis is to try to understand the role that microbial communities plays in the health, adaptation, and recovery of seagrass meadows. And specifically today, I'm going to discuss an experiment that is part of an ongoing study from which we just obtained our first microbial characterization results that allows us to ask some questions. So first of all, to go into the context, this experiment takes place in the Mar Menor, which is a coastal lagoon from the southeast of Spain. Probably most of you already know it, since it's one of the most unique ecosystems in our region. In general, coastal lagoons are transitional environments between land and sea, and they are characterized by high productivity and the great ecological and socioeconomical benefits that they provide. However, because it's semi-enclosed nature and the intense human pressure that it faces, Mar Menor is an extremely vulnerable ecosystem. But moreover, over the last decades, this lagoon has been suffering an intense eutrophication, mainly caused by the input of nutrients that comes from the intense agriculture or the urbanization of the surrounding areas. And this process had led to a lot of hypoxia or even anoxia events, the increase of turbidity, and therefore the decrease of light availability, and the loss of biodiversity. And all this ultimately result in the huge transformation of the ecosystem, as you can see in the video that you have on the top of the screen. So in this context, one of the most visible changes that happened in this lagoon was the disappearance of the Cymodocea nodosa seagrass meadows, which is a key habitat within the ecosystem for all the essential functions that this seagrass provide. And then we, in addition to the loss of Cymodocea, this lagoon suffered the significant extension of Caulerpa prolifera. This green macroalgae is native from the Mediterranean, but is considered opportunistic since it's able to spread really fast. And thanks to its high capacity to grow and develop in degraded environments, in the recent years has been colonizing most of the sandy bottoms of the lagoon and even areas that previously were colonized by Cymodocea nodosa meadows. As you can see in this map, that is from a study of our colleagues from Oceanográfico de Murcia. You can see clearly how the ecosystem was in 2014. I think this is not really clear, but basically we had mixed meadows and more presence of Cymodocea nodosa meadows. And then if you look of in 2016, right after the eutrophication events, most of the vegetated bottoms were lost. And then little by little, in the recent years, the vegetation of the bottom was recovered, but this time for Caulerpa prolifera meadows. So we changed from seagrass environment to algae environment. And with all that, given the importance of seagrasses and the regression that they are suffering, its conservation and restoration is a priority. But I think that first we need to understand really which are the factors that may influence their stability and recovery. And here is where microbes comes to the game. In terrestrial plants, it has been already shown that the microbiome can be an important factor influencing the resilience of the plants. And despite seagrass microbes hasn't been so studied yet, we know that these microbes play a role in seagrass health and develop. They create complex communities that are intimately associated with the plant for different reasons, as nutrient assimilation, as fixing nitrogen, or the uptake of phosphorus, synthesizing growth promoting compounds as hormones, or also detoxifying the environment by participating in redox reactions. And they also can protect the plant from pathogens or environmental stress. And now, yes, with all that, we can say that the microbiome can be considered as a functional extension of the plant that can play, can directly affect the resilience of these plants. Although the importance of the microbiome, just a few studies has been developed on this environment on the Mar Menor. So that's why we wanted to, in this work, analyze the differences in the microbiome associated to Cymodocea nodosa for different areas of the Mar Menor that have different environmental characteristics. So we asked some questions. We wanted to know if there are differences in the microbial communities across the different sides of the lagoon, and then if these differences are also different between different habitats inside or outside the meadows, and if there are differences, which are the factors that determine these differences. And then after that, we also wanted to try if we can use native microbiome to promote the success of transplantations in restoration activities. So to answer that questions, we carry out an experiment where we sample 3 sides of the lagoon: the north, which is the most open one, the center, and the south, which is the closer one. And in each one of these sides, we sample 2 habitats, inside and outside the meadows. I have to point out that our first idea was to sample vegetated and non-vegetated areas to be able to compare. But as I already explained, nowadays it's almost impossible to find any sandy bottoms of the lagoon since everything is covered by caulerpa. So when I'm talking about inside meadows habitat, we have mixed meadows of caulerpa and Cymodocea, and outside is only caulerpa prolifera meadows. Then in each of these habitats, we in both inside and outside, we take samples of environmental samples, sediment, and water. And then for the inside meadows habitats, we also sample plants, where we just take different compartments, in this case, seeds, roots, rhizosphere, and leaves. And these samples were used to analyze the microbiome by the sequencing of cystiner's gene. And also, in parallel with that, our colleagues from the Mar Menor, the IO de Murcia, were following the seed production and the germination from each of these populations. So let's go with the results. For microbe, we for now just analyze alpha and beta diversity to compare richness and composition of the community, the microbial community. So first, for alpha diversity, we have the environmental data here. The orange, because I don't know if you can see that right, the orange are the north side, the green is the center, and the south is the south. And we have the sediment samples and the water samples here. And when we analyze all the sample types together, we only find significant differences for sample type. So we decided to separate by compartments to can see if there are anything else any other differences, basically. So here we find for sediment samples in my right, for the habitats outside the middle, we find differences between north and south. And for the water samples, we find differences inside the middle for south compared to the other sides. Then when we compare between habitats, we find differences for water samples in the south, inside versus outside the meadows. And for sediment, we find differences in the north samples. For plant compartments, since it's true that we can see some differences in the diversity, these differences was not statistically different. So we can say that the richness stays similar for all the plant compartments. Okay. Now we know how is the richness, and we wanted to know how similar these communities are along all sides of all the variables. We have this plot. When we analyze beta diversity, we find that there were significant differences for all the variables, site, sample type, and habitat. I know that this graph is not easy to see. I just wanted to show again that the samples are mainly grouped by sample type. So you have the water in the side, the leaves, and then the sediment on the bottom. And here, more related to each other, the rhizosphere, the roots, and the seeds. But again, to be able to see it clearly, we analyze by sample type. And here we can see that the samples are grouped by sites, and north was the site that shows more differences in community composition compared to the rest of the sites for all the sample types. And here for the environmental samples, have the sediments here and the water here. We also can see that the square, which is inside the meadow, are a little bit different from the outside the meadow. And the same pattern in general, because I know that it's different for roots, we find that north was the community that more differs in metadiversity for all the plant parts. So, relating all these results to the environmental characteristics. Mar Menor shows the gradient of quality that goes from better preserved environments to more degraded ones. We have the north, that is the more open side to the Mediterranean, and we can find shallow and oxygenated water with sandy sediments. And also here we have better developed Cymodocea nodosa meadows. And about bacteria, we find more abundance of aerobic microbes in that site compared to the others. Then in the center, we have intermediate condition with fine and mixed sediments, also mixed meadows of Caulerpa and Cymodocea, and also the same for the microbes, a mix of aerobic and anaerobic microbes. Here we have more micro related to organic matter degradation and this thing compared to the north. And then the south was the more enclosed area with low oxygen levels and low light availability. Also, the sediments are muddy, and here the bottoms are dominated by Caulerpa prolifera. Of course, we also have Cymodocea because we sample here, but it's more difficult to find Cymodocea nodosa in that site since everything is covered by Caulerpa. And in this site, the bacteria predominant are the anaerobic ones, more related to sulfate reduction, degradation of organic matter. So in general, we can say that the microbial community is influenced by the environmental and biological conditions, and we can use the differences in microbiome as a bioindicator of the environmental status of the ecosystem. We find that the microbial composition differs across the different sites of the Mar Menor. And we also find that the well-preserved seagrass meadows can favor the selection of some microbes. We find that when we analyze the sample, there were differences between inside and outside the meadows in the north. And they have different bacteria, so it suggests that the plants create the environment that works better for her. So with that, I wanted to say that there are correlation with the preservation of the meadow and the microbial community associated. But we cannot say which is the cause effect. We can say that there are a reciprocal influence between the environment, the microbes, and the plants. So with all that, we move to the next step of the study. That is more related to seed germination and seedling development. Here we ask what happens if we change the environmental condition of the seeds produced in a different ecological context. And we also wanted to know what happened to the associated microbes when we change the plants. I will say that, I already mentioned that I think that our colleagues from the Mar Menor had been following the seed production, the germination, and development during our study. So they find some differences. In the center was super difficult to find seeds, which is also a response, but we took this community out of our experiment because it was super difficult to find seeds. In the south, the seeds were bigger, but there are not so many seeds. And in the north, we have a lot of seeds, but so much smaller. So we wanted to analyze what we call reciprocal transplants, which means that we took seeds from the north and we moved them to the south. And then we took seeds from the south and we moved them to the north. And of course, we also plant seeds on its own side as a controls. Without going so deep with the method, basically we just put seed plots in each side of each treatment. And 1 year later, we sampled half of these plots to analyze the development of the seeds, the morphological and physiological traits, and of course, the microbiome. And the rest of the seeds, the other half, were moved again to the other side. So the plots that were planted in the north were moved to the south and vice versa, because we wanted to go back a year later and took that plants again. And with that, we wanted to analyze what determines the success on germination and also what happened with the microbes associated to the seed. So do the seeds retain the native microbe or they adapt to the new environment? And also what happened to the germination outside the origin site? So what is more important for these seeds to, for example, find a place which is better conserved or they are ready to grow in the conditions of the native or from the mother? So if the mother has been exposed to one conditions, do the seeds need that kind of conditions to germinate? And I hope to be able to give answer to that probably in a year. But yeah, this is everything for today. So thank you.
Thank you very much. Xesca, any questions?
I have a question. So thanks for the talk. It was really interesting. A bit scary.
Yeah.
So, but the last thing that you mentioned of the plants retaining their microbiome or not, I was wondering whether it would make sense, and if it makes sense, whether you have thought about together with biological elements transplanted from the north to the south and vice versa, putting some abiotic bits, so piece of plastic or something else, to check what's the difference between the replacement that is due to just the fact that the new microbes are going to find themselves in a different environment, and a potential replacement or not due to the fact that the microbiome on top of the plant interacts with the plant and therefore might be favored with respect to the thing that is available in the new site.
Yeah, so just to make sure.
Does it make sense?
Yeah.
I mean, it was a little bit of contradiction. I don't know if it,
I don't know if I follow right. So you are.
So you're putting a plant from north to south, for example, and then you're going there and checking that, for example, there is a difference.
Yeah.
What part of this difference is due just to the fact that there are new microbes around, And what part of the difference, or maybe lack of difference, is due to the fact that the original microbes stay there because of the interaction with the plants. In order to test that, I was thinking perhaps you could also bring something that was colonized, a surface that was abiotic surface that was colonized in the north to the south and vice versa.
Yeah, yeah, we didn't thought about that. I mean, it makes sense. I think I will need to think deeply on that question, but my theory is that since the seeds don't have a huge richness of microbes, that when we change, probably the community will be that the plant will take the microbe from the area, because also these microbes are related to the condition. So maybe that's why you are asking to put a biotic.
But you showed that your beta diversity, there was quite a difference between the microbes in the water and the microbes in the seeds, which were similar in composition to those that are attached to the rhizome.
Yep, the seeds are buried in the sediment, so probably because of that, the water column is in movement, and the microbes are so different. So the microbes create a really intimate relationship with the part of what they are attached. So it's not only biological, also physical of the sediment and everything. So yeah, I don't really know how to. Yeah. I need to think more, because it makes sense. Yeah.
I have a curiosity about, about methodology on how you get, maybe you explained it, but maybe I missed it. Because the microbiome are the bacteria that are living on the surface of the leaves and the seeds and everything. How do you, how is the handling of the samples to, I don't know, for me, I see like super complicated, right? If you have something that is buried in the sand, taking the bacteria that are in top of it, but you have to remove bacteria from the sediment, I guess. You take everything together.
Yeah, for example, comparing sediment to rhizosphere, so the bacteria are super different. So we take the samples and we extract the DNA through a process, and we are taking all the sediment sample, and it's true that, for example, the rhizosphere is in the same environment, but the bacteria are different. So the bacteria are really attached to one part of the plant or the sediment. So for the leaves, the same. They are the bacteria that are attached. We just cut the leaves and las trituramos, and we extract the microbes, the cystinous microbes, and we remove also all the plant and other stuff that we are not interested in during the analysis.
So you clean, you need to clean the leaves? I don't know, because I don't know if there might be.
Yeah, for example, for the leaves, since they are on the water column, so they can be missing, we rinse the leaves with autoclaved seawater, for example, and the bacteria that are really working in the leaves and in symbiosis with that part of the plant are the ones that stay. So, yeah.
Another question? Okay, so we can leave it here. Thank you very much.